Uh, there's a lot of knowledge in the system on continuous manufacturing because manufacturing has been there for a very long time, and it's accelerated in pharma over the last 10 years. In Cipla, we've had the experience of moving on a hybrid continuous manufacturing, both on the API side as well as the formulation side. And therefore, on the talk today, I'm going to focus on Cipla's experience on continuous manufacturing and largely focus on the formulation side of it. And I'll request Nikita if you could play the video which I had circulated on our line.
To meet the healthcare demands, in India and world over, Cipla has introduced state-of-the-art automated, continuous manufacturing facility, which is regularly upgraded to meet current CGMP, safety, health, and environmental standards. Approved by leading authorities around the globe, dedicated to solid oral dosage. By linking all process-related machinery to servers, to record real-time process parameters, we have digitalized the manufacturing activities. Let's take a look into the flow of our continuous manufacturing process. With our staff member, going through the primary change room, for his gowning procedure to ensure that his body is fully covered from head to toe. It starts with raw material being received, weighed, and scanned. From here the raw material will go through sampling process, and later analyzed at quality control lab. Approved consignment is stored in automated storage and retrieval system. The approved raw material will be weighed and measured in the dispensing booth as per the process order requirement, whilst the dispensed material will make its way to continuous manufacturing unit for production to start. Our workforce needs to go through the secondary changing room to get to their respective stations. MES digitizes production process and guides through the production flow of the continuous manufacturing. The process starts with the preparation of raw materials, which are then fed into IPC, using the integrated sifting method. Then IPC, with the raw material, goes through the step of premixing, which is fully automated. Afterwards, the IPC is integrated into a single continuous line to process further the pre-mix blend. First stop, roll compaction, where it will form equally sized dry granules, by combining the powders containing the active components and excipients. Once the granules are formed, it will be shifted to continuous blending and compression, where it will be blended with other excipient with the help of continuous feeder and blender. PAT tool ensures real-time quality assurance. When the blend is formed, it will shift down to go through the process of compression to form tablets. The newly formed tablets are then put into check master machine to verify the hardness, thickness and individual weight of the tablets. Following the test, it'll go through continuous coating machine to achieve coating with an outer dry film to protect it against the environmental conditions and to get desired therapeutic effect. After every production process, it's mandatory for our IPC to go through cleaning procedure. Our facility is equipped with latest technology to ensure quality testing and training of our workforce. To support the continuous manufacturing unit, the facility has a packaging block. Additionally, we have a dedicated service area for smooth functioning of our continuous manufacturing line. At Cipla, we believe that our patients put their healthcare needs in our hands and rely on us for quality and affordable medicines. With responsibility and innovation, we are committed to fulfilling those needs and serving our purpose of caring for life.
Great, thanks. We can go to the next slide, please. So as I articulated earlier, we started the journey on continuous manufacturing on API in 2018. As we speak, we've actually developed 4 molecules through the CM route on API, of which 2 products are at a commercial readiness stage, and we're waiting for the respective patents, etc., to go off for us to put these products in the market. On the formulation side, we started the journey in 2023. Again, we have got two products ready, and we're in the process of commercializing them as we speak this month, right? When we started the continuous manufacturing journey, almost everybody talks about these three separate impacts that continuous manufacturing can have. First one is integrated processing with a fewer step, shorter process times, higher efficiencies, and higher level of safeties. The second one is essentially a reduced cost, smaller facility and equipment, reduced inventory, WIP, and flexibility in operations. And the third one is the ability to go for online monitoring and control for higher productivity and real-time quality assurance. So what I'll take you through over the next 5 to 7 minutes is, as we embarked on the project, what were the things which we considered and what are the results we actually got at the end of it. Uh, right at the outset, I would say that all the three goals we ticked when we completed the development. Let's go to the next chart, please. So when we were setting up the continuous manufacturing process for Cipla, we had a lot of brainstorming done as to what is the right process we should adopt. And we realized that end-to-end continuous is not something which we can look at, but we should go for a hybrid model so that we bring in where the technology has an impact. For example, we move from wet granulation to dry granulation and roll compaction. We moved to continuous blending, which would give us a higher level of output throughput. Compression, of course, is continuously fed and then followed through with continuous coating. So when you embark on this journey, there are 7 key elements which need a very careful consideration. In our case, we wanted to go for a process which will give us very large run times and volumes. So we actually have a capability to have 100 million pills batch size, though we have currently qualified it only at a 40 million batch size level. So we went for a dry granulation process in terms of the products which we shortlisted for this. Again, when you're selecting the product for the processes which you've identified, then you have to look at the kind of materials which you're going to use, whether the flow properties are right or not. How are you going to lay out the facility? Because one big element of this whole continuous manufacturing is our ability to shrink the footprint of the facility. And therefore, how you go about blending, compressing, how are you transferring material from here to there, whether you go for a vertical coating, horizontal coating, those kind of things. And then we had also a task to understand the equipment which we use, because these were essentially very, very new to us. And therefore, we asked the vendor to give us equipment on hire much before we embarked on the project so that we could actually experience the material, its flow properties, how it will respond to batching, what kind of quality qualifications it would need. So we did a complete 2-month pilot on the blender to understand the equipment and how we will take it finally when we go to the commercial stage. The other area which was quite important was in terms of the skill people have in terms of both technology as well as digital skill. And then because the batch sizes are too long, how do you go about sustaining the run? How do you do changeovers? How do you optimize on that? So those 7 elements are very, very important if you want to have a successful continuous manufacturing facility. Let's go for the next one, please. So when we were discussing and debating what to do, how to do, and what kind of product which we could do, unfortunately, given the file transmission, I cannot show you the videos which are embedded here. But we leveraged digital twins to ensure that we optimize the process much before we commercialized it. At Cipla, we developed digital twin for almost all equipment categories, whether it is mixing, whether it is blending, whether it is compression. So for all the 6 equipment which you see on the right, we have digital twin capability. We have digital capabilities also to monitor environment, do smoke studies in a smokeless manner. So we leveraged our manufacturing science and technology group to do a DEM simulation of the line, understand what parameters we will be doing, understand the space in which we will operate the equipment. So this helped us optimize the whole process in a big way. Let's go to the next chart. This continues. So we actually did simulation of large pitch blender, small pitch blender. We worked with the vendor, creating digital twin, doing in silico modeling of the process so that we optimize the equipment while even it was with the vendor, making choices in terms of pitch, all of that, so that the formulations would end up as a success. So a lot of effort went into the development of process, leveraging the 4IR tech, which we are all familiar with at this point of time. Let's go to the next chart. Same processes were also used in terms of defining the process parameter and the operating space, which we leveraged for the various product. And this was both the last chart and this chart, there was a lot of interesting discoveries because this would be roughly the first time way back in 22, 23, where we were doing in silico process modeling and leveraging these kind of technology for real-time optimization of process material flow, etc., and the output. So a lot of effort went into this kind of optimization, and the results will speak for it. Let's go to the next shot. So if you see, we ended up reducing the cost per thousand pills by 83%. It was in the band of 800 to 900 rupees per thousand pills. The final cost of manufacturing, we will optimize it further. As we speak, is lower than 150 or about 145 rupees per thousand pills. The facility which we put, which you saw in the video, that's the entire facility. And that can have an output of 2 billion pills in a year. So we actually reduced the footprint by 80%. So this is a complete tick. We got everything that we started with. We also were able to reduce our dispense to dispatch time on typically the OSD dispense to dispatch time tracks anywhere between 26 to 30 days. We've been able to bring that down to about 12 to 14 days with this facility. And the most important thing, what we've been tracking in terms of reduction of extent of touch as a part of our digital transformation journey. Most of the operations in the classic stage would have about 90% extent of touch. And what we typically refer to as a touchless operation tends to have about 55% extent of touch. And we've been able to bring this facility roughly in the band of 60, 62% extent of touch. So that's another box which we've checked. And along with this tech, we were also evaluating online monitoring control, real-time quality monitoring. So that is the box which also we checked. So all the three boxes which we identified as a objective for the CM journey, we've been able to achieve with, and the production is actually at commercial stage. So I must admit that CM, all the promises it makes, actually delivers that. I think that would be my last page, and we can go on to Q&A if anybody has. Yeah, that's right.
Sure.
And what we've done is quite similar to what I think Cipla's journey has been. So you will find some common thoughts there. And I think that is a good coincidence, perhaps paves the way for many others to speak to either of us. And hopefully we will see more such experiments in the industry. If you can please go to the next slide. So this is, a typical, now we do not have this. This is an experience center at a supplier. And we evaluated such a true continuous manufacturing line very comprehensively. And we thought there would be some time before, as an organization, but also as an industry, will be ready to take the leap to a truly continuous manufacturing process. We were lucky during AKEMA, we were able to visit one of the big pharma's actual continuous manufacturing line, which was under qualification, and hence our team could see the line. Now, this line was 8 years in design and qualification. Now, take out 2 years for COVID, but it's still a period of 5 to 6 years that was taken for the line to be designed. They began with vendor number one. It didn't work out. They went to vendor number two. And with the vendor number two, eventually the line was installed, qualified. And interestingly, what we noticed was that when we were visiting them, the head of the project, who was also the head of R&D, was in the process of moving on to another organization, right? So that suddenly 8 years of IP being lost. The other thing which I find a challenge is when you look at these big pharmas, right? You will have postgraduates in pharmacy or science or engineering running the lines as operators. Now, we have in India a dignity of labor issue, and if you were to ask a M farm or B farm or an engineer to actually operate the line, you may find a challenge. You may find attrition. People will think of it as not befitting their status. And then you risk the entire technology because these are all very sensitive lines, a lot of technology embedded in. So that for me is a challenge. At what stage to make that leap of faith in the talent that will actually drive these lines? The next slide, please. If you can just do a few enters here, so let the full slide rather than going into animation mode. So these are things which I'm sure everybody knows. I will not dwell on this, but I think the lines between scale up and actual commercial scales get blurred when you go into a truly a continuous manufacturing line. This would be an end state for us. Now, while all of you can go through the benefits listed here, and happy to talk about them in the Q&A, what we decided at Zydus was that we thought we will lend discrete manufacturing processes to a learning stage wherein we chose the continuous blending and coating as operations that we have started working on with respect to the continuous manufacturing. And our experience has been very, very positive, which encourages us to go to the next steps. If you can go to the next slide, please. Yeah. Now, the continuous blender, we have this from a European vendor, and this gets mounted right on top of the compression machine, which also means that in many of the current sites, the room height itself will become a limitation. Unless you're lucky to have an appropriate room height, the operation, whether you look at this or whether you look at another vendor who supplies a high efficiency blender which gets hoisted on top of the compression machine, the room height is indeed a practical limitation. We looked at two vendors which do this operation. The basic principles are the same. The continuous blending, the blenders themselves, there are only a couple of people in the world who manufacture these. One of them you saw, Garrick in Cipla, right? So in that video that was there. The loss of weight principle is what drives the continuous blending operation. We chose this for the MUPS product, wherein we thought that the KPI that we were chasing is product robustness, the cost being a byproduct. And we really wanted to eliminate batch-wise blending in this. And this will be also visible in the next slide. If you can go to the next slide, please. Yeah. So currently, when you look at, right, the steps from a granulation to compression will include a blending, batch staging, bin transfers. Now, all of you are aware more than I am that a bin transfer, batch staging, all of these can create issues with the delegate pellets. And then again, the issues like segregation, pellet damages, etc. And when you go to a continuous blending, you eliminate the top three stages and you go right from granulation to compression, with the blending happening right on top of the compression. Next slide, please. Now, what we did was in the trials that we took, a benefit that we were hoping for and we did get, was that of a much higher speed. Now, what we did was in the trials, we took multiple samples. We tried for 3 speeds actually. One was the current speed, then one was 80 RPM, and another was an intermediate speed of 65 RPM. We took the stabilization samples initial, and then we kept taking samples every 5 minutes. So the whole idea was to see how the blender behaved throughout, and also when the pellets were coming to an end, how did the sample and the product behave. Next, please. It's an interesting one wherein at 80 RPM, our AV was 11.1. It was better than that at 50 and 65 RPM. Now, it also therefore offers us a higher throughput from 50 RPM to 80 RPM. Right? That's a 60% upside in throughput. Again, that has benefits in terms of throughput and therefore cost per thousand. But what we were chasing really was good content uniformity, good product quality, least damage to pellets. And therefore, we were taking pellet samples at various stages to see whether the continuous blender, the blender itself could damage the pellets, whether they would get damaged at any point in time during the transfer process itself, especially when you're feeding the machine at this RPM. And we found no damages at all. Next, please. And as you can see here, the DISO that we achieved at various time points was good, and uh, this established the technology worked for us for the MUPS product. Next, please. You can please, yeah, sure. So what we found here is, like I said, there's a 60% upside to the throughput itself from 50 RPM to 80 RPM. That's 30 RPM above 50. The staging time is gone. There's a footprint that you're avoiding. There's a in-stage inventory that you're avoiding. So it's a very lean operation, and multiple stages have been eliminated. There's no quarantine, and you basically, with the help of the PAT technology, you have advanced in process controls. So therefore, you have much better control on the product that you are producing as the batch goes. Next, please. The next uh, area that we used the continuous manufacturing was in coating. Now we have established a plant with a very high capacity, a very high size batch size. We're looking at batch sizes of around 6 tons there. And in that facility, we are using continuous coaters. The rated capacity is 1200 kgs per hour, but we have been able to establish 850 kgs per hour throughput in these continuous coaters. And again, the limitation is obviously that they are suitable not for modified release product, but they are more suited for IR kind of products. So you basically, it's a very simple operation. There'll be obviously multiple zones within the coater, and the tablets move in at one end and come out at the other, simply put. Next slide, please. Now here, if I can, just establish the differences between the two technologies, the continuous coating and the batch coating. Your product contact time goes down significantly, which also means that the possibility of tablet damage is much less, because you have a product contact time of 10 to 20 minutes in the case of continuous coating, versus in a batch it could be 6 to 7 hours, even more in some cases. The first one obviously is a rate dependent process. The second one is time dependent. The handling is completely almost eliminated in the case of continuous coating. And you are free to therefore discard a certain quantity of coated tablets versus the entire batch being at risk. Again, no scale ups, etc. required. And therefore the development cycle is so we have been able to develop tablets of 6-7 milligrams right up to 1 gram coater. and that's been a good achievement. The discharging is automatic. The batch of the output per hour we have been able to achieve at 850 kgs, versus in a standard batch coating would be around 55 kgs per hour. And the spray rate again is significantly almost 40-50% more than the batch coating. Obviously, the limitation is that this is used more for IR compared to the batch coating, wherein you can use both for IR and MR. Next, please. Another study that we were able to establish is that a single continuous coater would comfortably replace 4 batch coaters. Now you can imagine the impact that it has in terms of whether lean, whether in terms of process robustness, process improvement, and footprint, manpower. So every which way, I think this for your IR product, this is entirely very commercially viable. I think the challenges for the industry would be striking the right balance between capex and opex. Our strategy is more around moving towards opex based decisions, and capex would therefore be more flexible in terms of how we take decisions in technology. So this was quite an interesting outcome, and it allows us to slowly convert wherever possible batch quotas into continuous quotas as a part of the life cycle management of the equipment. Next, please. I think one is whenever I have spoken to my colleagues, I still don't find a lot of adoption of these even I will not call this completely continuous manufacturing in the true sense, but unit operations being continuous, whether it's coating, blending, or compression, etc. So capex, I think, is one inhibition, but when you really weigh it against opex, the decisions become easier. Industries, the equipment manufacturers, their role in either loaning the equipment to the industry or setting up better experience centers in India could be an enabler. And for people to be able to go to these experience centers, dirty their hands. But again, we understand that taking even these trial batches can be an expensive process. So that's something which I believe industry can come together, conduct these trials together, and therefore dividing the cost of experiments across multiple organizations. And that could be a way forward. QA mindsets, which often can be inertia based, and also loaded with the regulatory concerns. How will the regulatory agency see? Plus, also, I think often you find that R&D is working on different technology and manufacturing, plus MS&T is working on different. So unless you embed these technologies right at the formulation development level, R&D level, you will obviously find that the new products are not getting developed on this technology, and therefore you are doing a full rework at a point in time with the product itself. We are committed to making this widely used across Zydus, and we'll be happy to collaborate with anybody else who's interested in use of this technology and share our experience with them. This is what I had to share. Thank you.
IPA for the opportunity. I think quite impressive to see the fantastic work that has been done in Cipla Zydus on implementing continuous manufacturing as a part of their operations. Today, what I'll take you through is our journey in terms of developing a fully continuous and connected manufacturing process for biologics. And I'm going to start with a little bit of a background of the company just to get you up to speed with where we are, but not too much time. But some of it is also important because I'm going to share some cost of goods data, which is related to the various scales that we have transitioned through. So if you go to the next slide. Next slide, please. Next slide. So we are a biotech company. We started in around 2016 in Pune. And when, so the whole genesis of continuous manufacturing really came from our need to disrupt the cost of goods for the proteins or products that we manufacture, right? When we started, there were a lot of companies ahead of us who had done biosimilars, launched biosimilars in India for almost 15, 20 years. Their plants were fully paid out. And, for us at that point to look at a business where biosimilars was our, level one business plan, um, we had to do things differently, just following what others had done with products already on the market and coming with a new biosimilar, which is using the identical technology to the 5 other companies who've been doing it for 15 years did not make sense. So, which is how the whole genesis of continuous manufacturing came. So, can you go to the next slide, please? And in terms of our brief journey, we started as I said, in 2016. We did our R&D. We developed a lot of these products. Again, when we were looking at technology, we implemented this in R&D first, and we said, okay, let's look at this differently. Let's come up with a completely different technology than what others are using from a cost perspective. Because for biosimilars, if you're not able to address the cost of goods, then you don't increase the access, and really there is no business, right? And so after we had developed it in 2017, 2018, we set up our pilot plant where we did our clinical manufacturing. And then we set up in 2019, 2020, our commercial manufacturing plant. We now have 7 biosimilars that are commercialized. We've done around 30 products. We're also CDMO. We've done around 30 projects for, all novel biologics for companies mostly based in the US. And so now we are setting up our plant in the US, which will also host continuous manufacturing lines. If you go to the next slide. So, when we started out, really our purpose was to democratize access to these life-saving medicines for patients in India. And next slide. And when you think about biosimilars as a business, this slide kind of talks about why cost of goods is incredibly important, right? So what this slide shows is the annual household income in India on the x-axis, and then the y-axis percentage of people who are within that income bracket. So if you look at, say, around $1,500 a month, it's only less than 2% of Indians really as a household who earn that, right? And then when you look at biologics, I think, India, of course, is a power center when it comes to generics medicine. But when you look at biologics, the problem is slightly different that needs to be solved. And the next slide will show you what that problem is. If you go to the next slide. So again, $1,500 a month, only 2% of Indians as a household earn, right? 2% of Indian household. But the cost of these products is extremely high, right? For example, just to correlate to the cost of gold, which everybody understands in India quite well, $80 per gram. While just to manufacture some of these monoclonal antibodies, just simply manufacturing costs, okay, can be up to $500 or so right now. If you're at a 2,000 liter fed batch, if you scale it up to say a 10,000, 20,000 liter, which is where some of the bigger CDMOs are, then you can probably bring it down to closer to $100 or $150, right? But this affordability challenge for biosimilars is real, in a sense, right? The cost of manufacturing itself is quite high and needs to be managed. And our whole intention for continuous manufacturing was to address that. If you go to the next slide. And when you look at biosimilars as a business in general, it is a low volume business because, these are patients who are critically ill. And so although, it is very important to provide access, it is a slightly different business than, say, vaccines, which are given to everyone, or some of the other generic medicines that India is done quite well in, where the volumes are very high. So when the volumes are very high, it is really your direct costs which ultimately start having an impact on the overall cost of goods. But when the volumes are low, so if you go on to the left-hand side of the screen, you can see the blue bar there, which is the fixed cost, which is going to have a bigger contribution to the final cost of goods, right? And the fixed costs come from, of course, utilities, maintenance, salaries, amortization. But the big thing also, of course, is the depreciation. And so these biotech plants are very expensive. And so when we started out, we said, okay, you invest like $200 million in this plant, which is going to depreciate, get loaded on the products that we sell. And at that point, it really doesn't make sense then, right? Because your cost of goods are going to be very high. So if you go to the next slide. So for us, the what we were looking at was to reduce the cost or reduce our investment into the manufacturing plant, and the best way to do that is to really shrink the footprint down, right? I mean, if you have a smaller footprint plant, then you'll have to invest less, and your operational costs are also going to be low. Now, what is shown here in this slide is a typical fed batch process, okay? And all the squares there, the orange squares, are the hold time. So in a batch process, after every unit operation, you have to hold. And for biologics, these are very complex processes because you're using live cells to make your products, and you need to have at least 7-8 different steps to purify it so that you get the right quality of product in the end, right? So typical monoclonal antibody, you will have all of these unit operations, right? And if you have to hold the unit operation, you need a hold time. So almost 60 to 70% of your footprint for a fed batch process is consumed by your hold time, right? So for us, it was very simple. More than continuous, we said if you connect all of these, then all the orange squares are gone, which means you have a very small footprint, and you can save a significant amount of investment that goes into putting the plant. Okay, so if you go to the next slide, so essentially that's what our initial. Can you go to the next? I think somehow the slide has not transitioned. Next. I think the previous slide somehow didn't go through, but essentially for us, it was about connecting all of this unit ops, which is what this is our R&D scale, which is shown here. So this took a lot of effort, actually, to be honest with you. The first time in R&D, of course, we were able to develop it, but once we went into our pilot plant, for the first time, and we connected this, because you have to remember the problem also is bioburden, right? Because you're using an extremely rich cell culture feed and media where bacteria can grow. Your cells will grow, but other bacteria will also grow, right? So all of this has to carry it out in a way where your bioburden is maintained very low. It's almost an aseptic process in a sense, right? And so it took us a significant amount of failures. Actually, 25 times we failed before we were able to successfully do it in our GMP pilot plant. If you go, can you go to the next slide? So the big thing that happened though, of course, the footprint was small, but then volumetrically also, how do you get the same kind of output that you need, right? And I think with perfusion, so the first step that happens in a continuous manufacturing of biologics is the perfusion, where upstream you're continuously putting the feed in and continuously removing the spent, what we call the spent media, which is the used media. And along with the spent media, the product comes out, right? So when you do that, because you're refreshing the media continuously, the cells are healthier. So what is shown on the left-hand side of the graph here is the cell densities. So generally, we end up getting somewhere between 7 to 10 times higher cell densities than a conventional fed batch. And if each cell produces the same amount of protein, then what you see on the right-hand side is a significantly improved volumetric productivity, right? So you get around, in this case, around 46 grams per liter from a 2-liter bioreactor, right? So we are not only actually making a smaller footprint, but we are also able to get a significantly higher volumetric productivity. If you go to the next slide, and that generally leads to somewhere between 50 to 80% lower cost of goods. So I'm going to show you detailed slides on cost of goods, so I'll not spend too much time on this. If you go to the next slide. So this approach, of course, works for monoclonal antibodies. In the next few slides, I'm going to show you a specific example of cost of goods and the drivers behind it. But in this slide and the next case study here, I'm going to also talk about difficult to express molecules, right? So monoclonal antibody is a platform now in biotech, biopharma in general, right? But the newer modalities, which are human engineered, are very interesting because they're very potent, but they are also very unstable, right? Anything that is human engineered seems to be unstable. So this is one client who was trying to manufacture this tri-specific molecule. They went to a CDMO in the US, 200 liters, but because there were proteases that were chopping their product out, if you go to the next slide, they were not able to really get too much yield, right? So what is shown here is on the right-hand side, the Kumasi blue gel on the right-hand side, most of your screen, you see all of these bands. So, 200-liter bioreactor at the end of 15 days, they were just getting the product completely chopped up. They came to us in 2 liters. We were able to manufacture more material for them than they were getting in 200 liters, because anything that is unstable, where in the 15 days of fed batch, the product is going to break. Continuous manufacturing, where you are continuously removing the product and bringing it in a stable state, of course, works extremely well. And in this case, if you go to the next slide, we were able to get 27 grams only in 20 liters of a perfusion process. So now we have INDs open. The client has INDs open for this, and clinical trials are ongoing in various different locations. But essentially, in this case, perfusion and continuous manufacturing was the only option, to be honest with you. If you go to the next slide. Now I'm going to show you a few, and next. I'm actually going to show you the cost of goods. We talked about the, can you go next slide, please? We talked about the, impact that continuous manufacturing can have on cost of goods. Can you go to next slide, please? I just want to, because for the sake of time, I want to go to like real data here. So if you remember, I had talked about a pilot plant that we had set up. So often we get asked this question in terms of what is the real cost of goods saving for a fed batch versus continuous manufacturing. So this is a real life example where we had 2 products, one which was in fed batch and the other one which was in continuous manufacturing. Both had very similar productivity from the cells, around 0.8 to 1 gram per liter, right? So it's kind of an apple to apple comparison. So now when you look at 50-liter scale for fed batch, the output was around 32 grams. As I told you, it was roughly around 1 gram per liter. And so the cost of goods at 50 liter will be $4,000 per gram. Okay. So 50 liters is clearly not an ideal scale, but we had a very small pilot plant from which we're trying to make this clinical material, right? So, but as you scale up, of course, this will come down. But now when you compare it to 40 liters of the fully connected continuous manufacturing process, you can see that we have almost 5 to 6 times more output, and the cost of goods even in the 50 liter is 1,000, right? So you essentially have almost reduce the cost of goods like to like at the same scale by 70 to 75% lower when you go to continuous manufacturing. And what is very interesting here, on the right hand side is, if you look at the direct to indirect cost ratio, your indirect cost is of course very high because 50 liter is not an optimal scale, right? But even at this scale, when you go look at the bottom portion of it, your indirect costs are still manageable, right? When you do perfusion, because of the higher output that you get. Can you go to the next slide? So now we took this CMAB and we scaled it up. So this was just a 40 liter. We scaled it up. Can you go to the next slide? No, just one before this. Sorry, there seems to be a lag. So now here we are going to take the same molecule and we are just going to go up the scale. Of course, the manufacturing plant has changed. So, the indirect costs are slightly higher because this is a bigger footprint now. And as you can start seeing, when you go to 80 liters, you are at $533 cost per gram. And then when you scale it to the 320 liters, which essentially is the same suite, you're just scaling up the bioreactor and the entire process. Now you've gone to $147 a gram, right? And this is only in a 500 liter bioreactor. Now, if you run 500 liter fed batch, you're going to be probably more than $500, $600 per gram. So again, with perfusion and continuous manufacturing, even with the clone, which has a lower productivity, you are at a decent cost of goods of 150. And as you can see on the bottom half, your direct to indirect cost ratio is now being more ideal, right? I mean, generally, you want to have your indirect cost to be around somewhere between 20 to 30%. That's when that you are operating at an optimal scale. So in this case, even with the 500 liter, we are reaching there. Now, if you go to the next slide, so now what we'll show is, now this is a model for the same molecule. We've modeled, what I think I don't know if the bottom is visible to all of you or not, but essentially, the third bar on the from the right is the 500-liter scale, and the fourth is a 1,000-liter scale, right? So you can see that as you scale up from 50 liters to 500 liters, you get a significant advantage of just the simply economy of scales. But beyond that, for a productivity which is around 1 gram per liter in this case for a cell line, beyond that you're not going to get a huge jump with scale up, right? So now we are at $147 per gram for this particular cell line, and if we really need to go to that $50 per gram, we need to have higher productivity cell lines. So if you go to the next slide, this slide just highlights that, right? So the newer cell lines are a lot better in terms of productivity, and so we can expect almost 50-60% increase in the productivity just because the cell lines are better. If you go to the next slide. So with these higher producing cell lines, now we are at around 10 to 15 kilograms per batch, and at this stage we are around $70 to $80 per gram, right? Just by increasing the cell output and the overall productivity. If you go to the next slide. So of course, we are taking the same platform, and we want to now implement this in the US. If you go to the next slide, so we are coming up in the US with a similar platform in a sense. And if you go to the next slide. And this just shows you where our cost per gram is going to be. So with the current platform, we are around $77 when we scale it to 1,000 liters. So that scale up has happened in India to 1,000 liters. It's ongoing right now. And from US, we'll be around $96 per gram. And if you go to the next slide, because the operational costs are slightly higher in the US, and now with NZX 2.0, we are actually implementing process analytical tools. We are looking at higher titer clones, which we already have developed in R&D. And we are also further optimizing the cell culture media, because 40% of your cost of goods is still in the cell culture media. So any development there will help further reduce the cost of goods. So if you go to the next slide. And this just shows you where we are in terms of cell lines. So now we have cell lines, which actually in 22 days here, we are getting 29 grams per liter. When you stretch it now to around 30 days, we are essentially crossed the 50 grams per liter barrier. So we are currently at around 60 grams per liter with these cell lines. Previously, all the data that I have shown you so far was around 20 to 25 grams per liter in a month. Now we are at around 60 grams per liter per month. So just by that, I think when we scale this up, we will be able to take the cost of goods even easily below 50 or $40 a gram. If you go to the next slide. And the real benefit of that through our partners is really for the patients, right? Because unless you use technology to further harness lower cost of goods, I think with biosimilars in India and for novel biologics for early stage products in the US, it's very difficult to further improve the affordability. And it's really technologies like this that will allow us to really further break this barrier. So actually, that's the end of my talk.